Hi guys, my name is Emilio Vasquez, and I want to share with you guys a little science fair project. So maybe you're an overachiever, and you think if I do this science fair project, I'm gonna have something really good to show on my resume. Or maybe you're someone who likes science, and you're in it for the experience. Or maybe you're none of these two people, and you're just doing it because you heard your chemistry teacher say, "Who wants extra points?" Well, if you're none of these people, or somehow all of these people, I want to share with you guys this little idea. Right before Thanksgiving break, my buddies did a science fair project, and I thought it was really, really cool, so I filmed every single part of it, and I'm here to show it to you guys. So without further ado, let's show you the project, what you need, and what you can expect. Let's roll the clips. Alright guys, so I want to present to you guys a biology project. For this, you're going to need 3 1-gallon containers, some rubbing alcohol or Germex, 10 sterile nutrient agar plates, we found these for cheap on eBay, 16 test tubes with a sterile nutrient broth, our teacher was kind enough to provide us with these, and pipettes with pipeters, we found these for $8 on Amazon. And of course, as with any good science fair project, you're going to need some gloves too. This is definitely not a project that you want to do the day before science fair projects are due. This thing takes at least 2 to 3 days. However, if you're planning on being a biologist or a microbiologist and you want to work out in the field, this is definitely a very eye-opening project where you get to see and experience what these guys do on a day-to-day basis. So with that said, let's get on with the project. What are you guys doing for science fair?
Okay, so for science fair, we're gonna collect effluent water, which is also called waste, from this river that goes down Central, Robindale, 802, and South Padre Island Boulevard. We're gonna collect 3 samples: one before the PUB plant, which we're gonna collect it here, and after the PUB plant, we're gonna collect it right in by Robindale, and 1.5 miles down, we're gonna collect it in the South Padre Boulevard, which is also by the Port of Bronzeville.
Alright, so out of these water samples, what are you going to test?
We're gonna test bacterial growth between each site. We gathered water and then we diluted them into 1/100,000th, and then placed them into agar plates where we would grow the bacteria into colonies.
Alright, so out of these sites, what do you expect to find?
Well, since there's a lot more sewage being dumped into the river before the PUB plant, we expect there to be a lot of colonies and a lot of major growth right before the PUB plant, and right after, we expect there to be minimal to almost none growth at all between the bacterial colonies. And 1.5 miles down in the port of Brownsville, the reason why I did it 1.5 miles down is so that we give the bacteria a chance to grow again and for more bacteria to be added into the river, and we expect minimal growth between it, between the point after the PUB plant, and that is what we're gonna test.
Alright, guys. Well, good luck on your science project. So we got to the first site, and unfortunately, that day was really windy. There's just a lot of wind, and the video quality is affected by it a lot. You can tell it's obviously really pixelated, it's really shaky, but not much I can do about that. I'm sorry, guys. Anyways, so when we first arrived there, we tried to throw the empty gallon jug down with a rope attached to it, and that proved to be a really massive fail. So we got down there and we kind of just submerged the gallon jug down under the water. And remember, if you touch any of this water, don't touch your face, whether that be your ears, your eyes, your nose, any of that. This is wastewater, it's really dirty and it's really disgusting. So after you get the water and after you clean your hands, you're going to want to seal these up with the cap, and you're going to want to label these as well. Um, you can bring in a Sharpie if you want, or you can label them some other way, but we used a Sharpie. And you're going to want to place these jugs in an ice chest, and this is so the metabolic rate of the bacteria inside the water is slowed down. And what this basically does is that it keeps the bacteria from using up the nutrients that are inside of the water with them, allowing us to get better, more accurate results from the actual test site. So after we got all of our jugs, we placed them in the ice chest, we labeled them, we took them back to the lab, and we're going to show you exactly what we did right after this clip. There's a shopping cart. There's a friggin' shopping cart, and this water looks the cleanest, guys.
And we got the most.
It does. The water looks the cleanest here.
So weird. Well,
Yeah, and there's a tire right next to
this
it.
place is whack.
Maybe deceiving
Whack.
though. Alright, I'm gonna pour a bit out.
Ooh.
So after getting our water from the field, we went back to the lab test tubes, our sterile auger plates, gloves, and our pipettes and pipeters. However, our teacher ended up giving us some glass pipettes and a real pipetter, so it's going to look different in the video as opposed to the $8 plastic ones I referred you to on Amazon. However, it's going to work the exact same way. You're only going to have to be a little bit more careful when you're taking the measurements because it could skew the results if you take too much or too little. Anyways, it's also important to note that every time you transfer liquid from one container to another, you're going to have to use a new pipette. This is just to get more accurate results. Let's say that leftover bacteria or leftover nutrient broth don't go into the next vial, into the next test tube even, and they skew further results. Anyways, so with our test tubes, we're first going to label them S1, S2, and S3. Every site is going to have 5 test tubes, and we're going to have one extra, and that's going to be our control. So we're going to label these numerically, starting with 1 over 10, 1 over 100, 1 over 1000, 1 over 10,000, and 1 over 100,000. And we're going to repeat this for every site. Obviously, not the control though. Anyways, so we're going to take 0.5 milliliters from the first jug, for example, and we're going to put it in the test tube labeled 1 over 10. And after that, we're going to put the cap back on, and if you don't have a cap, you can put some very clean plastic film on it. You don't want to contaminate. And here, we see a young scientist out in the wild. Notice how this young lad does not have his glove on. It is also important to note that this lad got very sick the next week. Unfortunately, 2 days later, it was also Thanksgiving, and he couldn't have any of the bloody turkey. Stay safe, kids. Be smart. The results, of course, and you're gonna shake it up very well. The more you shake it, the better. But don't overdo it either. It's also important to note that if the solution is not shaken up enough, then the rest of the experiment could also be affected. So, after we're done shaking it up, we're going to take 0.5 milliliters, just like the first time, and we're going to place it in the next test tube, the one that's labeled 1 over 100, and we're going to repeat the process. You're going to do this until you reach 1 over 100,000, and you're going to repeat this for jugs 2 and 3. For your control, however, you will not add anything to it. At the very end, with your control, you're just going to take 0.5 milliliters of it, and you're simply going to put it on your sterile auger plates. For your other test tubes, the one that actually came from sites, you're going to get 0.5 milliliters from the 1 over 1000, 1 over 10,000, and 1 over 100,000, and you're going to place them into their own auger plate. Remember, it's very important to label these plates and not lose track of which plate is which. Let these guys incubate for at least 24 hours, but no more than 36. And if you want, you can also record how much exact time has passed. So, all of your data will be observational, as you can't quite accurately measure how much bacterial growth happened inside the auger plates, since it's a very minuscule amount of growth and they don't really weigh much, so you can't weigh their biomass too effectively either. So most of your data is going to be observational, and some of your data may even come from your nose. More on that from the results. So after around 27 hours, we took the bacteria out from where we were incubating them in their auger plates, and we opened them up to get a closer look inside. For this segment, I only took a picture, and even though a video would have been nice and all, the smell from the bacteria was just way too much for me to handle. I was covering my nose with my left hand, and when I wasn't covering my nose, I was just coughing, and so I ended up just holding my nose the entire time. And even when I was holding my phone with my other hand, it didn't leave me with many ways to open up the auger plates and give them a closer inspection. So I decided to just take a picture, and I made sure that this one and only picture I took was at least clear for you guys, and I hope you guys really appreciate the effort that went into me standing that close to the auger plates. So I hope you guys know what you at least can expect from the results. If your results don't look like this, it's okay. Results will vary depending on your location, on where you got the samples from, etc, etc. So, I'll see you in the outro. Alright guys, so for now, that'll be all from me. I hope you enjoyed the video, and if you have any questions, leave them in the comments. I'm on my phone pretty often, so it shouldn't take me too long to respond to many of these. I did want to apologize for that last segment one more time, but if you go through all these directions and you do it correctly, you're going to more than understand why I couldn't just stand being next to that bacteria for as long as I did. But for now, that'll be all from me. I'm Emilio Vasquez, and this is your science project. Stay frosty, guys.